Hi everyone, and welcome to the refresher session for General Biology 1. My name is Elisa Septuche, and I'm going to go over just some brief topics from General Biology 1 that will help you in this semester. So first we're going to go over what macromolecules are. Macromolecules are the building blocks of life. They are composed of two parts. One is called a monomer. A monomer is a small organic molecule, so just one single molecule. And in order to help remember that, mono means single. And then there's polymers. Polymers are it's like a long chain of multiple monomers. So it's like, for example, you have one single Lego, and then if you were to connect a whole bunch of Legos that look alike, you will have that long chain of Legos. That's kind of what a polymer is. So it's multiple units of monomers put together. And those are going to make up 4 different classes, which are carbohydrates, lipids, proteins, and nucleic acids, which we'll go over in the next few slides. First, we're going to go over carbohydrates. Those are going to be sugar molecules that you take in. So carbohydrates are monomers. They are classified by monosaccharides, which are simple sugars that contain 3 to 7 carbons. So let me get my laser pointer. So this right here is a monosaccharide, and it says single sugar molecule. So that's going to be your glucose, galactose, fructose, etc. Then you have your disaccharides, which means 2 monosaccharides. So it's going to be 2 of these, which we see here. And in order to remember that, I always like to think di means 2. So for example, that's going to be your maltose, lactose, and sucrose. And then you have your polysaccharides, which we learned from the last slide that poly means many. So it's a long chain of monosaccharides, which can be branched and unbranched. And for example, that's going to be your starches, glycogen, and cellulose. So these are going to be your really big carbohydrate chains. And this is going to be what a starch looks like. So when it says branched, that's what it means. And then unbranched are like fibers, which is this. Next, we're going to have lipids. So lipids are organic molecules that are insoluble in water, which means that they don't dissolve, or they're also hydrophobic, which means water fearing. So those don't like to mix in. So think of like when you mix oil and water, you can see the little clumps of oil forming as it goes into the water because they don't want to be mixed in. So fats are large lipid molecules that are made up of a glycerol head and a fatty acid chain, which are used to store energy. So this is going to be your fatty acids right here. This is your glycerol head and then your fatty acid chain. Cells are going to be made up of phospholipid membranes, which have a hydrophobic tail and hydrophilic head. So over here, you have your plasma membrane, which is what all cells have around them. Then you have your hydrophilic head, which does like to be in water, means that likes water. And then you have your hydrophobic tail, which doesn't want to be mixed into water. And when it is mixed into water, this is what it looks like, because it does everything it can to keep the water out of these tails. Now you have your examples of lipids, which are going to be fats, oils, waxes, phospholipids, and steroids. Next we have proteins. So proteins are built from amino acids and form a 3D structure. Some types of proteins that are used are enzymes, storage proteins, receptor proteins, transport proteins, contractile proteins, and defensive proteins. A protein shape is specific to its function. So certain, all of these have different shapes, and even those have specific shapes, like each category. So there's tons and tons of proteins in your body, and no two of them are exactly alike, but they are going to have the same build, I guess. So they start as an amino acid, and then that's your primary structure. Then you have your secondary structure, which forms a helix of those amino acids, and then you have your tertiary structure, and then your quaternary structure. So that's just a simple breakdown of what a protein looks like. Lastly, you have your nucleic acids. So the example here, DNA is a nucleic acid. It stands for deoxyribonucleic acid. DNA is broken up into 3 parts: a sugar group, which the sugar is called deoxyribose, a phosphate backbone, 4 nucleic acid bases, which are adenine, thymine, guanine, cytosine. So those are all the parts of DNA. This is going to be your sugar phosphate sorry, not your sugar, your phosphate backbone. This is just holding everything together. These are your nucleic acid bases, which make up your DNA. And then you have the sugar, which are the white molecules right here. That's your deoxyribose. So yeah. So here I have just added some additional resources. They're going to be covering the central dogma, which is just DNA to RNA to protein. And where is it? So he's going to discuss the difference between DNA and RNA, and then DNA replication, transcription and translation from DNA to protein, and then mechanisms of DNA damage and repair. So this is one of my favorite educational YouTube channels because he does a good job in explaining explaining topics in short videos and doesn't make them too long. So you're going to see these links throughout my presentation. So now we're going to go over the parts of the cell. There are 2 different types of cells. The first one is prokaryotic and the next one is eukaryotic. So prokaryotic cells make up the domains bacteria and archaea, and then eukaryotic cells make up everything else, such as animals, protists, fungi, and plants. Those are going to be these are going to be discussed in the next few slides. We won't go into archaea that much. Just know that those are prokaryotes. So on this slide, we're going to discuss the difference between eukaryotic and prokaryotic cells. So a prokaryote is a simple unicellular organism. They are single-celled, which is what unicellular means, and they lack a true nucleus and membrane-bound organelles. They store their DNA in a nucleoid, so it's just kind of like loose inside the cell, versus eukaryotic, where they have a true nucleus where it's enclosed and protected. They are surrounded by a capsule, cell wall, and plasma membrane, versus, for example, in an animal cell, we don't have a capsule, we just have a plasma membrane, a cell membrane. Or in a plant cell, they have a cell wall surrounding them, and then the plasma membrane. They will always have ribosomes inside. So that's about what makes up a prokaryotic cell. A eukaryotic cell is more complex and it is multicellular. They must have the following 3 to be considered a eukaryote. It must have a membrane-bound nucleus. numerous membrane-bound organelles, and several rod-shaped chromosomes, which are going to be inside here. So these next two slides are going to be it's a chart that I put in that you can use to go back and reference if it is present in a prokaryote or if it's present in a eukaryotic cell, and if it is it an animal cell or in plant cells? And they also give the function of the cell component. So it's a very detailed and useful slide. So go ahead and take a look at these when you get a chance. I have also added some additional resources here to go over in more detail the prokaryotic cells, what those are, and then eukaryotic cells part 1, which are animals, and the endosymbiotic theory, and then eukaryotic cells part 2, which goes over plant cells. And just for a quick reference, I have also added this table that I found that in the book, and it's about the components of the fluid mosaic model. So it talks about which part of the plasma membrane and then what those parts do. So go ahead and look over that whenever you get a chance. So here we have different types of transports through the plasma membrane, because since the plasma membrane is what is used to connect the outside of the cell to the inside, or not connect, but like protect, there are types of transports that are used to get objects and molecules and nutrients and all kinds of molecules through and inside and outside of the membrane. So I will link a video in the next slide to go over it in more detail, but just as a quick rundown, you are going to have two types of transports, or three, sorry. You have passive transport, which doesn't require any energy to accomplish a movement across the membrane. So this is going to be your easiest. It's going from a high concentration gradient to a low concentration gradient, and it just moves with the flow. That's going to be simple diffusion, facilitated diffusion, osmosis. Then you're going to have your active transport, which requires energy in the form of ATP to move across the membrane and moves against the concentration gradient. So you're going from a low concentration gradient to a high concentration gradient. So you're going to need that energy in the form of ATP to get the molecules where they need to go. So you have primary active transport and then secondary active transport, and then you also have bulk transport. That's going to be like your phagocytosis and trying to expel waste from the cell. So on this slide, you're going to have your methods of transport, energy requirements, and types of transported material. So these are going to talk about the different types of transport methods, the energy that's used, and then what it is that those can transport. So go ahead and take a look at that whenever you get a chance. I also added some links here about the different types of transport and membranes and transport study materials, and this links to I think it's Khan Academy, and it has like quizzes and articles and little bits of information that you can use. Next, I also linked some videos that I thought were very helpful that cover ATP, cellular respiration, and photosynthesis. So all of this is going to be in that chapter regarding metabolism and what ATP is. You're going to have this video going over glycolysis, and then the next video goes over the citric acid cycle, which are all things that you need to know. And then you have the electron transport chain and oxidative phosphorylation, and then you have photosynthesis here at the bottom. So all of these tie together, so make sure that if you have any trouble in these chapters, refer to these videos or ask me for help. So now we're going to get a little bit into genetics. We're not going to go too in detail. We're going to keep it as brief as we can. But first, mitosis is part of the cell cycle. And the cell cycle is the events that cells grow and reproduce. The cycle regulates the growth of the cell to make sure that they are healthy and functioning properly. This cell cycle makes sure that there are no cancerous cells, no cells that are not functioning properly, and if they are, they will undergo like a self-destruction kind of mechanism. Then you have, so now let's get into a little bit of mitosis. It's the process where two daughter cells are formed from one parent cell. So this is making a clone of the cell. There's no different types of like outcomes. It's just one cell splits exactly into two, making two clone cells. That's all it is. You're gonna have prophase, and then you have your I'm trying to point but not get in the way. So you're gonna have your mitotic spindles. They're gonna be on the sides. You have your centromere in the middle of the sister chromatids, and then you have little tiny fragments in the nuclear envelope. That means it's getting ready to kind of like disappear. Then you have your prometaphase. Now at this point you have the spindles at the north and south part. They've already migrated apart from each other. Your kinetochores, the microtubules that are pulling those kinetochores apart, and then now you're getting into metaphase, which means that all of your kinetochores are going to be in the middle. Meta starts with M. I always like to think M middle. So now they're going to start to pull apart here in anaphase. So you can see where they're pulling those apart. And then they're forming the daughter chromosomes. They're no longer they're no longer part of the parent. And then you have telophase and cytokinesis. And all that is just this this little cleavage starts to form, which is where they start to split into two cells. Your nucleolus starts to form again, and you have a spindle on each cell. So they're going to be exactly the same as this cell was in the beginning. Now you just have two of them. So just to keep it very brief, I'm also going to have videos linked to this, but we'll get into meiosis a little bit. So meiosis is the phase that occurs when two gametes come together to form a zygote. This happens in sexual reproduction. Meiosis is like mitosis by repeating the same phases 2 times. So you're going to have your prometaphase, metaphase, anaphase, telophase, cytokinesis, 1, and then you're going to do it all over again, and then you're going to make your, so this is meiosis 1. You went from 1 cell to 2 cells, and then again, you're going to go through it again, prophase, metaphase, or prometaphase, metaphase, anaphase, telophase, cytokinesis, and from that one, you're going to make 2 more. So in total, you'll have 4 cells that are all different. They're not going to be identical. And this only occurs with sex cells and during sexual reproduction. I linked some videos here that can clear up a lot of confusion because I know meiosis is a lot, to take in. So I have a video that's about the cell cycle, and it talks about the different phases of the cycle and how it regulates cell growth. And then I had a video that talks about mitosis, how 1 cell becomes 2, meiosis and gametes and the human life cycle. Then I added a little bit here about genetics and Punnett squares to give you that practice, and then chromosome structure and organization to help you understand a little more about chromosomes. So I hope this it's a very brief overview, but I hope this helps. If you have any questions, please email me. My email is ezertuche, ezertuche 12@alamo.edu. I am in the STEM Center, and I will be more than happy to help you and go over some topics that I went over here, or if you have topics that I didn't go over but you want more help, please let me know. I'm always there for help. Send me an email or just walk into the center and look for me or check my schedule. It's always posted, but please reach out to me. Yeah, so I hope this helped. Thank you. Bye-bye.